Yeah, hello everyone. I think I'm online. Yes, hi. Hello and welcome. Microbehunter here. And it's Saturday again. I cannot believe it. Time's passing so quickly.
Yeah,
Welcome to another
hello everyone. I
Microscopy
think I'm
live
online.
stream today. Let me
Yes,
sort out
hi.
like every time a
Hello and
little
welcome.
bit
Microbehunter here.
essentials. And it's Saturday
Okay, I
again. I
think
cannot believe it.
seems to be
Time's
working, I
passing so
hope.
quickly. Welcome to
Okay,
another
hello from Vietnam, from the
Microscopy
United
live
States, from
stream
London.
today.
Isn't it beautiful that so many people from all
Yeah,
around
let
the
me
world start to
sort out like every time a little bit
Northern Germany.
essentials.
Okay.
Okay, I
Live
think
stream
seems to be
153.
working, I hope.
Hardly
Okay,
I
hello from
counted it.
Vietnam, from the United
Thank you.
States, from London.
It's
Isn't
unbelievable.
it
I've been
beautiful
doing
that
this
so
now,
many
what, for
people
the
from all
third
around the
year?
world start to
I cannot believe it that
Northern
actually-
Germany.
Yeah.
Okay.
Good evening from England.
Live stream
from
153.
Germany,
I
from
can
northern France.
hardly
I'm
uncounted
echoing.
it.
Ooh,
Thank you.
echoing.
It's unbelievable.
thank
I've
you
been
for
doing
this
this now what for
info.
third year.
Let me- I know why
I
I'm
cannot
echoing.
believe it that actually. I think I should not be echoing anymore. Okay. Yeah. Yes. The reason is why there was an echo is because I've got several cameras connected, and there was a second camera that also picked up the sound and kind of mixed it in, and that's why there was this double echo thing. Okay. Yes. So I switched off the other microphone. Luckily, I knew what the problem was because some time ago I already had the same problem. Okay. So it seems to be working again. Okay. Echo is gone. North Carolina. Okay, fine. Thank you very much for telling me, because I do remember a couple of months ago, years ago, actually, for whatever reason, there was absolutely no sound at all. So you probably already know, if you've joined in already last week, that I've changed around the format a little bit, and instead of talking about one topic, I've kind of decided to split up the live stream into several smaller topics. Gives it a little bit more variation, and it also makes it easier for me, because it's easier for me to talk about several smaller things. And for today, what I want to do is first of all, ever smaller Jesse sent me a picture, CD picture, which I'm going to show you. Then talk a little bit about the new format. I want to mount a mosquito. Indeed, I was, believe it or not, breeding some mosquitoes. Or at least they appear to be mosquitoes, because I collected a water sample and they hatched. So that's something I want to do. And I bought a different, look at this, what I got here. This is a fountain pen ink of different colors, and I want to test them to see how well they are suitable or not suitable for staining my cheek cells. So I've not tried this. Okay, the only thing that I know is that the blue color works, but I did not try the other colors yet. So this is something that I wanna experiment a little bit, right? I did buy myself just now this USB camera. I already had a similar one before, but when I'm going on holiday, I want to take it along, maybe to do a little bit of microscopy. Yeah. Then I want to talk about this magazine here, which unfortunately is not available anymore, because there was a little bit of an interesting article that I found in here that I just want to share with you. So I want to talk a little bit about this magazine as well. And then there is a viewer choice, snake skin, or this moose. Remember this one from last week? And most viewers decided not to look at it. And today I have got some snake skin over here. Let's have a look. How many people actually want to look at which one? Yeah, you still have a possibility to. Snake skin is winning. Yeah, okay. I'll put this under the microscope. I have to tell you the following. It's keratin. Keratin is a protein, right? So this is actually not, to my knowledge, it's not made of cells a lot. It's mostly protein that kind of is shed off. There should be actually some cells as well. But let's have a look at this, okay? I'll tell you then later also a little bit how I got this snake skin and the background a little bit. Let me, And then I'm going to do, again, a Q&A session. As you can see, I've reduced it a little bit in length, okay? So I'm quickly reading every now and then, yeah. Yeah, so that's indeed that's basically water-based fountain pen ink. I have to say the following: some of the inks are certainly a mix of different pigments, right? So I can imagine that the purple one is a mix between blue and red, maybe. Yeah. So these are not going to be chemically entirely different. It doesn't matter. I've not tried it yet, and I would simply like to experiment a little bit with that. So that's a little bit the plan, what I have, and I simply want to take it from there. And the numbers that you see here, that's approximately the time that I intend to spend approximately on each one of them. Okay, I got an email the other day from Ever Smaller. Jesse, and here it is. There were actually 3 pictures. I just want to share with you one of them. So thank you very much for giving me the permission to share this. And maybe you can, if you want to, you can also maybe comment a little bit on here. This here is a Ever Smaller, also right now in the chat. This is a CD-ROM. I think it's pretty cool. I tried already to make a microscopic picture of a photomicrograph of a CD-ROM, but I could not see the pits, those dashes and dots so easily. So this is actually pretty cool, I guess. I don't know, maybe by illuminating it a little bit from the side. But honestly, I've never seen it with a light microscope in such it's I think quite nice. I'd like to try it as well. I think with a stereo microscope, definitely not good enough. But this one, I think it was 40 times and 60 times, and I think, and then contrast enhanced. Yeah, so I think it's that's pretty interesting that we're actually able to see, a little bit. Yes. Yeah, exactly. A Euromax 1153, 40 times, used a bright flashlight from the side. Okay, that's. Thank you very much. So that is something. So epi-illumination from the top, and then maybe if you're shining it from the side and it reflects in a certain way. That's actually, I think, also how the lasers actually work. Depending on whether there's the pits or not, then the laser is reflected differently, and that's actually causes. That's the way how you can actually read it out. So that's actually quite nice to see that with a regular light microscope. With the proper illumination. The only question that I have is because with 40 times, I mean, the distance between the objective and the CD is quite small already. So I usually had some problems sometimes because sometimes the objective is casting a shadow, and then it's sometimes the specimen surface itself is not fully illuminated. At least that was one of the problems that I usually had with higher magnifications. But that's It's a very nice thing that I haven't tried it yet, but I'll also try to do that. Yeah. So if you, by the way, if you have any, if you want to send me a couple of pictures to share with the community, please feel free to do so. Yeah. I read off right now. Yeah, it was difficult. That's why the 60 times photo was so much worse. I had difficulty getting the light under. Yep. Yeah, because the distance between the objective and the CD is so small, and then there's always a shadow, so you have to have a really shallow angle to actually make sure that you illuminate it. Yeah, so that is basically so if you have any also pictures that you would like to share, it would be really nice. Desk view. So let's talk a little bit about this mosquito thingy here. So it was like this. New format. No, I'm not finished yet. I want to talk a little bit about this. 1.5 to 2 weeks ago, I started to change the format around of my 2 microscopy channels. So right now, the channel that you're listening to this right now is called Microbehunter Microscopy. And a couple of years ago, I used to do live streams here on this channel. Then I switched over to my other channel called Microbehunter. And I continued to do live streams there. And now last week I switched back again to this channel. And the reason is because I wanted to separate I've got 2 different types of audiences. I've got an audience that's mostly interested in the hands-on microscopy stuff that I'll be doing today. And then I've got another audience who seems to be more interested in the more general, general microbiology stuff, right? So I want to separate the topics a little bit. So in case you're wondering, it's going to be like this, that most of the hands-on microscopy stuff will now be published on this channel here. I do intend to publish approximately once a week an explainer video, and I've already got the first one ready. And I'm going to publish this then also maybe 4 times a month. I don't know exactly yet, so I have to establish a system, but I intend to publish approximately once a week an explainer video, which is going to be scripted out a little bit. It's more the traditional style of video. Please, what I'm going to do after the live stream today is I'm going to put in my other channel, I'm going to put the finished video online, and it's going to be it's going to be very different in style. It's going to be a classical explainer video with some b-roll on top. Please have a look at it, okay? I'd like to hear your feedback in any case, okay? So after this live stream, I'm going to put it online in my other channel, in the Microbehunter channel, okay? It's going to be a little bit about the Cassini space probe, space, and what it has to do with microorganisms, okay? Are you sure you're still teaching? I will be honest with you. Marie is asking this to me. I'm lucky in the sense because right now I've got summer holidays. Okay, yeah. So that is this is the thing. I've got a summer, and that's why I do have a little bit more time right now. So let's do the following. Yes. Could we create a cross section of a fly or a mosquito? We could, but I'd have to plan this ahead because this would actually mean embedding, okay? And it's something I have not done yet, but it's something that I also plan to do. I'm going to make a whole mount of the mosquito, okay? So this is, I need to explain what I have done. If you watched one of my videos in this channel a couple of days ago, I published a video about a bird bath and about the rotifers and the Hematococcus that I found in the bird bath. This was in this channel a couple of several days ago, maybe I think last week. And this is the water sample. And interestingly enough, there were also some mosquito larvae, or what appears to be mosquito, in there. And they hatched. So I put a lid on top. There is enough air in here. And those mosquitoes came out after 1 day or 2 days, and several of them. And I kept the lid on top until they kind of died by natural causes. And then what I've done is I used a tweezer and picked them out carefully without trying not to damage them too much. And I transferred today in the morning, in my time, so several hours ago, I transferred the mosquitoes into alcohol. So what you see here is more or less concentrated alcohol. And there are 2 reasons why I've done that. Several reasons. Number one, first of all, it removes the water from the mosquitoes, and therefore it dehydrates them, and therefore they're more, how do you call this, permanent, right? When I make a permanent mount, because I want to make a permanent mount. And the second reason is it kills off bacteria, because otherwise those bacteria would start decomposition. It's also something that I don't want. So let's do the following. Let's give it a short try. This is now the stereo view. And let's simply have a look under the stereo microscope, how those, let's make it smaller a little bit, how they actually look like. Yeah, you see they're fairly dark. Yeah. Echo again, really? Is there again an echo? Yeah, I believe. Why? Now the echo should be gone because it switched over to another camera, and therefore this other camera's sound turned on. Okay, now the echo should be again hopefully gone. But thank you for yeah, good. Okay, no, I know the problem because I switched on another camera. So that's basically how they look like, right? And what you see over here, this funny thing here, that is the leftover of the how do you call this? The pupa, or where it actually came out. Yeah. So I've got, I don't know what, 3 or 4 of these mosquitoes, and the thing is that they're fairly dark. Actually, they're quite black and not very transparent. Yeah, so that's a little bit the problem that I'm having here. So, and for this reason, when I mount them, the other problem is that they're fairly thick. That's another problem. So these are not ideal specimens for making a whole mount. Okay, I'm well aware of that, but I still would like to try it, and then hopefully after maybe several days or a week or 2 weeks, I hope that they will become more transparent, so that they're easier to observe. Okay, so yeah, so it's yeah. See, you see this guy over here is one of them has actually lost a wing. Yeah. So it's probably, these are not, just as for your information, these are probably not the nicest, these are probably not the nicest, how do you say, slides that I'm going to make. Yeah, but I just want to look at them, yeah, and see how they look like, and therefore I'm still going to make a slide, okay? So let me, if I can even open this. Okay, here we go. I need a cover glass. They're over here. And another thing that I need to buy myself, and I don't have that, is these tweezers are not suitable for picking up insects. There are special tweezers which are much more delicate and much more flexible. This is very hard, right? But they are very inflexible. But there are actually special tweezers out there that are designed for picking up highly delicate objects. Okay, so what I'm doing right now is I'm going to take up some of the sample. Okay, the mounting medium that I'm using is again the one for insects. It's the Uperol. By the way, some days ago I also made a separate video where I'm simply talking about different types of mounting media. So that is now the mounting medium, and that's the nice thing, I can transfer the mosquitoes directly from alcohol into Uperol. It is compatible, right? I could not do that with water, right? But here, and now that, gee, where are my glasses? You see, I'm already getting old. I need my glasses so that I can actually see what I'm doing. And I don't know, I'm just gonna go for this guy over here, hoping that I'm not gonna destroy it too much. Okay, with a lot of patience. Here it is. It's actually on here. Yeah. And here it is. Okay. I have to wipe this a little bit, otherwise there is the problem that yeah, and I'll do this also with alcohol. There's a little bit of alcohol here, and let's remove the mounting medium. And then, of course, a cover glass goes on top. By the way, I've not mounted them before yet, so this is usually always a little bit of a trial and error as well. So this goes on top. Sometimes there is, if this doesn't spread evenly, sometimes it will actually push the specimen to one side. Yeah, but this seems to be okay. It is a fairly thick specimen, right? So this is also not ideal. And usually when you do that, it usually always kind of, destroys the specimen a little bit. Sometimes wings break off or legs break off or whatnot. Now I have to actually wait for 6 weeks, 8 weeks maybe. But at a low magnification, it's going to be fine, right? I always keep on repeating myself, I know, but you have to be really careful that when you use higher magnification, that the mounting medium does not touch the objective, right? Yeah. So let's put this under the microscope. Let's change over and let's hope that there is no echo. Let me see. No, I hope there shouldn't be any echo. Let's flip this out. Let's remove and I have to find the thing now. Ooh, yeah, not very nice. This is, as promised, a pretty dark. Yeah, no echo, good. Yeah. See this, actually does this even still have a head? Look, legs broken off. But the point is the following. Let's just remember how dark it is, Okay, because I would like to actually demonstrate that maybe after a couple of days or so, maybe next week I'm going to show it to you again, there should be a significant clearing. Basically, it should become much brighter. Yeah, this is a pretty bad. I mean, I'm not able to see the antenna, which are actually quite nice. So you know what? Well, next slide. I'm just going to make the next slide. This is good because this is not really, yeah. So the desk view again here. So let's try another one. I am checking occasionally the time because I do want to stick to the time. So let me see. Maybe let's try this one here. Maybe that's the empty. This takes a lot of patience. Yep. Here it is again. I think this might be the not the. Yeah. And you know what? Just for the fun of it, let's because we have enough space here. Let's try another one. Let's save it a little bit. I mean, I don't know, maybe there are some folks out there who are actually doing this as a, how do you say, some biologists who are doing this as a job with the purpose of simply doing a biodiversity assessment, because nowadays I read there are now new species of insects, obviously, also appearing. So that's the second one. And let's try a third one. I don't know. This usually does do a little bit of damage to the insect. So third one. Okay. Yeah, I mean, just by looking at it, it does seem to be quite submerged, but it doesn't hurt to put a small drop on top. Yeah, this is a lot. Yeah, and cover glass. Here we go. And the specimen is quite thick, obviously, and you see that it doesn't spread, so it's probably going to take a fairly long time, for it to dry. Doesn't matter. Yeah, we're just going to have a look at it. Yeah, and there are a couple of comments here as well. Would you not look at it under the stereo microscope and spread it out before you put? Actually, it's also a possibility to do a little bit of an arrangement. That's what actually also one of the things that I thought. You take some toothpick, you try to arrange it. Oh, do I have one left here? I actually have, I don't know, maybe one or two left. One. I don't know. I could give it a try. Yeah. So let's have a look at it under the, how do you say, under the microscope again. Let me see some comments. Next week, a cliffhanger. Yeah. Yes, I will use Epi. I will put it under the stereo microscope as well. Okay, just. Oh, okay. This is now the, how do you say, the empty. This one actually looks quite okay. This one is now the empty skin out of which the mosquito appeared. There's a bubble in here, but actually this one is already quite transparent. This looks also fairly okay. Yeah. So this is basically the larva, yeah, will undergoes metamorphosis. And this is kind of the empty skin. And you know what? I'm going to go down with the magnification here. Now by 4 times I have to find them now where they are. Oh, okay. Here, that one actually is better. What about this one over here? It's also better. Let me see. I don't know how many I have. Yeah, but actually they look actually better. Okay, but you see it's still very dark, right? Yeah. So let's go up with the magnification. Yeah, and let's open up. You see, even if when I open the condenser, it's still so dark that yeah, I'm not able to see any details. But normally let's go down here with look at the antenna here. And then usually by looking at the mouth parts, we can actually see if it's really a mosquito or just some other if it really has this blood-sucking mouth parts or not. Actually, it doesn't appear to have those. Yeah, because a real mosquito has those long, but this one doesn't seem to have those long mouth parts. Yeah. Potassium hydroxide or hydrogen peroxide, yes. This is also something that I have done before. I've done this with ants, by the way. Some time ago I made a video about that as well. I put ants under hydrogen peroxide and I warmed it up. And this actually within a very short time caused a clearing of the exoskeleton. Right, so indeed it does make it transparent. I've not tried KOH, I've not tried potassium hydroxide, but I know that with hydrogen peroxide it does work. It does bleach it. Yeah. And there is a comment over here that about connecting a camera. I will picking up with a dropper pipette. Yeah, you know what? I could try that as well. Yeah. So let's do the following. They could be male. Males don't bite, maybe. Okay. So I'm going to do now the following. I'm going to take this and let's have a look under the stereo microscope. With light coming from the top. Okay. And as a matter of fact, that's how I made the thumbnail picture of this for this live stream. Yeah, you see, it's also a mosquito. And what I've done is I don't have it now here. I've actually taken a flashlight. Yeah, but you see, it's still very dark. Yeah. So and that's light coming from the top. Yeah. You see, it's somehow so dark that even that is not going to help out a lot. However, I do also have this handheld microscope, and you know what? Why don't I just try it like that as well? Let me quickly check a little bit. Let me check. Okay, yeah, 15 minutes, 10 minutes. Let's see, 50. Okay, now I'm just checking my time. Yeah, I should be actually slowly finishing up anyway to move on to the next one. But I will quickly do the following. Let's have a look. So I'll be using this thing here. Let's give this a try. And does this work? It has just yeah. Here we go. Did they just move? Yeah, they moved, but do you know why they move? Because I touched the cover glass. Okay, they didn't move on their own. But you know what I consider a little bit interesting, funny? It actually looks better. It looks better with this simple USB microscope than with my stereo microscope. Can this actually be possible? That's kind of interesting. Yeah, it's alive. No, it's luckily it's not alive. I accidentally touched the cover glass, and this caused everything to move. Yeah, but actually, you see, even with those little USB microscopes, it works surprisingly well, actually. Yeah. Yeah. Okay, I'll talk a little bit more about those here. Yeah. So that's basically the one over here. I'll quickly show this to you later. So let's go back to the desk view, and what I would like to do now is I promised myself that I'm simply going to simply interrupt myself with wherever I am, and I'm just going to always move on a little bit with the topics, so that I stick to this plan here, right? So about 15 minutes. So, and the next one is I want to try the different colored inks. So we are going to revisit those mosquitoes again, with the hope of that they will become more transparent by next week. Okay, so that's a little bit the idea. So let's put them away. I have to store them horizontally. And now let's do the following. Where is this? Here it is. Yeah, I bought myself several of those ink cartridges. Okay, yeah, they're a German company, I guess. I don't know, Pelikan. Green. This one over here is red. This one is it's black. Here, I mean, the color over here is purple, but actually it's a black one. This is violet, so it's purple. And this one is turquoise. So it's kind of a green-blue, right? Again, I guess that some of them are actually a mix of different colors, so they're not going to be chemically completely unique, but yeah, probably also mixes. But I would like to know how well they are actually good in staining. You're going to notice where's the blue one, the standard blue one, and the standard blue one is over here. I've got it over here in a separate bottle because I've been using it for staining. Now, there is one thing where I have to say where this is a problem, under quotation marks. This stain, the fountain pen ink stain, is not temperature stable. So if you, for example, put a drop of this blue ink into hot water, it's going to turn clear. Okay, so that's a how do you say? I guess deliberately done. If you get some stuff on your clothing that when you wash it with hot water, then it's going to disappear. Right? But it does actually work as a readily available stain, the blue one. And they're basically the way that I do that is the following. Is I will take some of the cheek cells again, and I will apply a little bit of this stain, but I will have to dilute it down approximately 1:1 or 1:3. Otherwise, it's going to be too dark. And what we should actually see is we should see that the cells will accumulate and absorb the stain. So the cells should actually become darker than the surrounding, right? But we can only see that if we dilute it down a little bit. So let's just get started, okay? So I collect a couple of the cheek cells. Question, are these inks suspensions or solutions? They are solutions, not suspensions. Otherwise, I will quickly explain as well what the difference is. Okay. So I'm going to apply it a little bit here. So a suspension would be when you have solid particles, for example, coal particles, carbon particles in water. That's a suspension. Sand in water is a suspension, and they don't work because these are then physical particles that are actually not able to enter the cells. And solutions are actually, just chemicals, so to say, which are small enough that they're also able to enter the cell. You know what? I'm going to go on. I'm going to, here I've got one of them here, right? I'm going to do another one. No, they're not specific for cell organelles like ribosomes. Okay, I'll talk about this as well. Because they basically, stains generally work if a stain is, let's say, acidic, then it will tend to stain those parts of a cell that are more alkaline, and vice versa. If you want to have highly specific staining, if this is what you want, really, you only want to stain, then you actually need fluorescent antibodies. Then, folks, and it's getting really complicated. So this is the thing, because chemically the different parts of a cell are not so different from each other. Okay, so I've got made two. How many do I have? 2, 4. Yeah. Oh. Let's. India ink and also certain calligraphy inks are suspensions. So there are certain inks out there which are for calligraphy, and they are suspensions, and they actually contain carbon particles or other particles. Yeah, and those particles are too big and cannot be taken up by a cell. Here, here. So I think we've got enough cells now on here. And now the trick is as follows. I need a pipette. Here it is. I need a fresh tip. And I'm going to do it the simple way. I'm just going to add water, a drop of, I don't know, shall we use 50 microliters? Let's just use 50 microliters each. Yeah, and 1, 2, might be even a little too much, I think. Okay, so what I have done in the past as well, I've done some heat fixing. I've made already videos about that as well. I'm not gonna do that. Okay, simply for the sake of keeping everything simple and just testing out whether it actually works. So, and now the fun starts. I need a system now to kind of remember what I have done where. So, number 1, I'm going to now, this is going to be now the messy part. You know what, let's do the following. Let's do the blue one first. And what I usually do is I just go in here and take a small amount. Okay. And you mix it a little bit. Okay. It might appear to be very blue here, but under the microscope it should be fine, I hope. So I need a, how do you call it, a trash can to exchange the tip. The next one, yeah, look. This one is now I need to get the ordering correct. This one is now the one, the turquoise. Up. Punch, I open it. Oh, it looks quite similar. So I don't expect a big difference here. You know what? What about labeling the slide? How is that? Blue. Turquoise, TQ. You know what, I'm gonna add TQ, turquoise. So the next one, I really wonder. I really wonder. The next one is what? The next one is violet. And black. You know what, I'm also going to label those here. TQ. Yeah. So the next one is violet. Up. Yeah, this looks already significantly darker. Next one is black. Honestly, I've not tried this yet. So this is gonna be interesting. Let's put those finished ones away. B for black. Up, look at this mess. I love it. You see that I'm not always taking the correct, the same amount, but it's gonna be fine. Red and green. Red. Red is easy because red is red. I can see that it's red. Look, I need to clean this here. Otherwise I'm mixing the stain. This was better. Oh, this seems to be very bright. No, you don't see it. Here it is. Red. And the last one is green. I would be very frustrated if none of them work properly. So, okay, here we go. I also label this here as a G for green, but what am I supposed to do with those cartridges? I have to probably throw them away anyway. So, and now we do the following. We now just simply add a cover glass to each and remove any excess. Here, maybe we don't even need to. Actually, maybe we don't even need to. Violet. Now you can actually see a difference in color. What was the next one? I forgot the next one. Violet, after violet, black. It's kind of B for black. Red, obviously, and G for green. Red and green. Yeah, actually looks pretty nice. Okay, so let's give it a try, and I hope that I've got enough cells on here, yeah, to actually be able to see something. I don't know. Okay, let's have a look. I'm a little bit nervous, yeah, because I don't know what to expect. So where is the scope view? Okay, so that is the blue one. Yeah, we do see something. But maybe it was a little too dilute, I just think. So that's the blue one. Yeah, normally the cells stain darker blue than this. But I think we can give it a pass. Okay. They do stain. Yeah. So the next one is turquoise. And there's a little bubble. And look, I think we have an answer. This stain is not suitable. Because you see the cells that you see over here. Okay, the cells that you see over here, they are not staining. Let me put, yeah. Okay, so yeah, look. So these are all cells here, right? They're not staining. Look, compare it to the blue one. Yeah, actually, yeah, some of them don't stain, but actually those that actually picked up, yeah, maybe because I did not add enough of the blue, yeah, ink. Yeah, at least they stain. The blue-green one does not. Yeah, so I think the conclusion, yeah, So I do not have that good point. Gives a nice background color if you don't have DIC. Yes, that's correct. I do not have DIC right now. Yeah, I have taken out the DIC. Yeah, so this is a regular bright field. So there is no no optical color change. Okay, we've learned something. I've learned something. Yeah, that blue works. The blue-green, the turquoise one does not. Well, at least we know that. So let's move on to the next one. The next one is violet. Let's see if the violet one, I go down with the magnification again. This is, yeah. The violet one also seems to work. Could be maybe that there is the same or probably the same pigment in there. So this one also worked. You look, it's actually quite nice because you can, let's go up a little bit here. You can actually see the nucleus as well of the cells. See that the nucleus actually do stain a little bit darker. Yeah. Most soluble dyes have a hard negative charge. Using a bit of acid might positively charge. Could be. I did not try to. I did not try to do pH adjustment. In any case, the violet one seems to work. Let's have a look at the black one. And that's the black. Look. Oh, okay. Just a second. Look. I mean, maybe I could have used a little bit more, but black also seems to work. You see, I think not all of the cells have stain. Maybe that's a question of concentration. But I think black also works. Look. Oh yeah, interesting. Definitely. Now you also see again here that the nucleus is much darker. So far, violet, blue, and black seem to be fine. It might be worthwhile to measure the pH of the inks. Yeah, I don't have such a small pH probe, unfortunately. So, and you know what? I'm gonna. Where's my pen? So this is a plus, which means it works. Yeah, you're still seeing the, how do you say, the microscope view. The next one is red. Let's have a look at the red one. So it's too bright. Red. The red ones also seem to be staining darker. Look. Yeah. I'm actually surprised that the background is that red as it is. But you see, the cells are staining darker than the surrounding medium. So red also works. I'm going to give it now a slight try. And then the next one is green. I don't know if it's... Let's go up a little bit with the magnification here. I don't know. You see, not quite as... yeah. Also still pretty transparent. Yeah. The frame rate is very low right now. That's interesting, really. Yeah. So yes, but the question is about washing the slide. I will try to do this now as well. Okay. The stage camera. This stage camera over here. Okay. I know that for whatever reason it generally is slow. Yeah. As you can see when I pass it, yeah. This is yeah. Unfortunately, I think this is it's always been like that. The problem yeah, I don't know why it is the case, but that was actually one of the few cameras that I was able to get which has a wide angle and also a manual focus. But you see, it's indeed quite slow. Yeah, it's for both, also for the one for the stereo microscope. Yeah. Okay. So, okay. So what I'm gonna try to do now is the following. Let's try to very briefly. I do want to, how do you say, always keep a little bit the time also in focus because I do want to talk quickly about other things as well. Let me quickly do a math 40. Yeah, 30. I'm already a little bit over time. Yeah, doesn't matter. I don't want to stress myself either too much. But if you want to now see if the pigment remains, what you do is the following. Look, you just take some tissue paper or filter paper, yeah, and you simply try to remove whatever stain there is on one side, and you add water. Okay, you try to add a little bit of water on the other side. Up. And let's go a little bit further down. And if you're lucky, then the cells will kind of stay where they are. Okay. Yeah. But the surrounding stain is removed. And if the cells have a high affinity for the stain, they should stay in that color. Let's simply try it again, okay? If this actually, no guarantee. Yeah, but let's hope that cells are now still red if there are any cells left. Scope view. Yep. So here we go. I think that's, you see, this is much better now. Yeah. Here the cells are now significantly, let's turn it up. Yeah, exactly. So yeah, you see the background is now more white and the cells are basically still red. Okay. So that's basically one of the ways how you can try to increase the contrast. Yeah. Red also stains the nucleus. Yes. Yeah. So you have with blue this also works. So this is actually a possibility of yet increasing the contrast. What you can try to do is simply put the stain directly, the concentrated ink directly on the cells, but then everything's going to be too too dark, right? And you still are not able to see a difference. But then you have to remove it carefully by using a tissue paper and removing the ink and then adding a little bit of water from the other side. Yeah. So we could just as a last demonstration, a very quick one. We could try it also over here, and then I want to move on to the next topic. I don't know. Maybe we can try it here with this one. You take and you just basically do it like this, and you add a little bit more. And look, doesn't this look nice? Yeah. Okay. Let's try it again. So. Let's wait a little bit until the tissue paper has removed. So it's almost, I think, yeah. Let's take away a little bit some of the liquid here from the side as well. Or why not do it like this? I don't like to have the cover glass float around too much. So let's see if there's still some cells left and whether they are still black. So where is the scope view here again? I have to find the cells. Yeah, here they are. Yep. Actually, the nuclei also look kind of nice. Yeah. Okay. You get the idea. Yeah. So in summary, blue works, red works, violet works, black works. The green colors don't seem to work. Like for example, this blue, green, turquoise, and also green, they don't seem to work. Yeah. Just for the fun of it, this here now is phase contrast. How does the whole thing look in phase contrast? Yeah. And you see that, yeah, the contrast is also quite nice. Yeah. So phase contrast also has its advantages that you don't need to use chemical stains. Are Reinberg filters an alternative for this ink? I would say probably not. Well, or maybe. If dark field works, yeah. You see, they're so transparent. The dark field doesn't always work, and if dark field doesn't work, then Reinberg would also not work. Yeah. So this is a little bit the issue that the difference in refractive index has to be sufficiently high, and if they're too transparent, then you're not able to redirect the light, and then you don't have dark field, and you also do not have Reinberg illumination. So that's a little bit the problem. Yeah. Why is it the inks that didn't work? Well, because inks have a certain chemical composition, and it depends on whether they're able to react with the different cell components or whether they're able to enter the cell, right? So that's essentially the thing. It's a question of chemical reactivity. And if they are made of a chemical that simply does not react with the biological components of the cell, then it doesn't stain them, right? And in this case, what we see is we see that the cells, once they're stained, they like to retain the ink, they like to retain the pigment, and this means that the pigment has actually bonded or reacted with whatever substances are inside the cell. Yeah, doesn't even have to be necessarily always a chemical reaction. It could be that the ink is maybe slightly more alkaline, then it reacts more with the acidic parts. Yeah, for example, DNA is acidic, so maybe that's the reason why the nucleus is more stained, because it's more acidic. Yeah. So dark field again. Yeah, but the problem with dark field, I only have 4 times with dark field, right? That's my problem. This means that I don't even know over here what are the cells and what's the dirt. Actually, I can show you. Do you see these things here? That's a cell. Yeah, you see, they're fairly transparent. All of the other dots that you see in the background is dirt and dust in the optical path. So dark field is very, very sensitive to that. Right? But you see that those cells here, they do not look very bright. Yeah? So I'm kind of concerned a little bit maybe that the cells are too thin or too transparent to allow for dark field. And if you don't have dark field, you also don't have Reinberg. Yeah, so this is a little bit the concern. Yeah, okay, people. You know what? I want to move on. I've learned now something as well. Okay, so actually those inks are useful, and if you don't have those, I don't know what, carmine or whatever. I can imagine maybe they are actually carmine or something like that. Could be maybe in here as well. Yeah. Yes, time. Yes, Robert is already. Thank you, warning me that I want to move on, and I'm gonna actually move on. Desk view. What is the next thing? A very quick intro into the USB camera, which you have already seen. Okay, so I'm making, by the way, a separate, more detailed review video of this camera, but I will quickly tell you a little bit the reason. Let me put the slides away first. The reason why I bought this, because I'm going on holiday, and I want to take a microscope along. Thank you for the questions. Okay, well, thank you. Let's see how far we are actually able to get. And the thing is the following. I wanted to take a microscope along, and now, yes, this is not a replacement for a, under quotation marks, normal microscope, but it can be a little bit of a replacement for a stereo microscope without the stereo view. I already have a similar one with a pretty bad resolution. This one is indeed better. Looks the same, and it allows me to simply look at things like this. It has a built-in LED. If you push it several times, you can actually adjust the brightness. There is also a little thing here where you can adjust the brightness over here as well. You might not be able to see it quite well in the video, right? The thing is the following. I can connect it, of course, to the computer, but I can also connect it directly to my Android mobile phone. This means I can take it along and can, I don't know, look at things also over my mobile phone and to record videos and make pictures. That's the bottom line. This here is annoying. 40 times to 1000 times. No, this is nothing more than a focus. Okay? Focus knob. I quickly want to show it to you. Let's do the following. It takes now a second until it starts to switch on. I do it like this so that you're able to see it, right? That's my beard. Isn't this beautiful? Actually, you can actually try to hunt for face mites as well, right? Yeah, and then you can, of course, where is this here? Yeah, this is now without any light. Now with LED. Yeah, you can actually adjust this a little bit and then refocus. To change magnification, I mean, that's the highest one. I have to move away. I get a lower magnification and I have to refocus. Okay? Unbelievable that this is actually how skin looks like. Yeah. What about the eye? Okay. This is how you can actually try to find face mites. Yeah. You can try to figure out whether certain spots, on your finger or on your hand are melanomas or not. I don't know. You get the idea, right? So you can do some casual nature observation using these things, and that's one of the reasons why I bought it, and just to have a little bit of fun. Yeah. But again, my suggestion is if you already have a compound microscope, if you have a compound microscope already, and you think about getting a stereo microscope, but maybe you don't want to have spend so much money for a stereo microscope, but you still want to play around a little bit, then this is kind of one of the things that you might want to consider. So that's basically just something I just quickly wanted to share with you. Okay, that those things are kind of fun to use. Not a full replacement, obviously, for a regular microscope, but I guess kids are also going to have a little bit fun with those. Yeah, it comes along also with a little stand. Yeah. So if you want to, I mean, look, some of you probably have already seen the video that I published a couple of days ago. I did not use this one here, but I used my regular stereo microscope. And look, there's these old coins. Yeah. And you can, and I was actually quite positively surprised about the fairly good image quality here. Ever small. I got it mostly because it's much smaller than a full stereoscope. That's correct. Yeah. So that is, and honestly, I don't know. I mean, if you don't need a super high magnification, I mean, it does its job, and it's fun to use, and I think especially in the field of hobby microscopy and just nature exploration and so on, if you find a tiny insect somewhere and you want to know what is it, or if you I've been bitten by ticks already before, right? So this is a little bit like a glorified magnifying glass, So. Exactly. Aaron Berg, I lead an ID mushrooms. This looks like a monoscope, looks useful for showing people, certain features. Also, for example, if you're doing some presentations maybe, with students, and you just want to show them something, and then that's actually much easier. Just wanted to share this with you, that this is basically my, my travel microscope, so to say. Not extremely flexible concerning, but it does its job for what's here. Okay, so desk view. So let's turn this off again. The only thing is that it did not come along with for the Android mobile. iPhones, unfortunately, doesn't work. But for the Android mobile, it did not come along with its own software. But you can actually download either free or paid viewer software, which are also able to record. And the free versions, I tried 2 of them, was not happy with those. But then I think for €5 or so, or €6, I paid for an app, and this one works actually quite nice. Yeah, so I can simply plug it in into my mobile phone, and then it'll work. Okay, so that's it. Then I'll move on because time is going. Yeah, one thing I want to show you, something interesting, fun, or not fun. Actually, it's a comment of one of my viewers who got me is the following. If you've watched, maybe you've watched, some of you have probably watched the video about the slide of the monkey's tail that I published several days ago. It's actually one of my favorite slides. It's the cross section of a monkey's tail, and it's an antique slide. And I like it a lot because it's very educational, but there was a comment, which you can read, by someone, and the following, and I just want to share with you the comment of one of my viewers, because this one actually is something that kind of got me started doing a little bit of research. So this was the comment here, the tail of a monkey, right? So if you have not seen the video, it's a couple of minutes long. It's in this channel a few days ago. Yeah. Hopefully the tail wasn't cut off specifically for the specimen. Okay. Yes, different times mean different customs. Thankfully, our values have changed as well. What was once commonplace can be shocking today. Yeah. For example, and this is now where fun starts. For example, I recall reading an article from approximately 100 years ago in the German Microscopy magazine Mikrokosmos. Yeah. All issues are available online. Yes, link is in the description of this video. They actually put all of the issues, they scanned all of them and made them available for free online. A valuable resource, even if you do not speak German, because you just get them automatically translated. That provided instructions on how to euthanize, kill, a domestic cat to prepare histological specimens from its organs for thin sections. So basically an instruction on how to kill a cat. Yeah. Nowadays, this would be unthinkable, pure horror. Manners and morals change over time. Let's be glad. I totally agree. Of course, I had to try to research this, and I found the article. I actually found the article, and it's horrifying. So I want to share it with you, and I did a translation into English. Okay, and I also want to use this a little bit to talk about this excellent magazine, Mikrokosmos, which unfortunately is not published anymore. I guess for financial reasons, they stopped publishing it back in 2014. So 11 years ago, they stopped publishing it. It's a pity, but again, all of the magazines are available online for free. And I actually found the article by doing an online search. Okay, here we go. And look, this is what I found. I printed it out. And this is basically from the year 1921-22. This is just the cover. Yeah. Yearbook of Microscopy, a magazine for applied microscopy, microbiology, microchemistry. I don't even know what microchemistry is, and microscopic technology. And indeed, there was an article in there, quite a lengthy article, which I also printed out. And don't worry, I did translate this somewhere. Where did I put the article? Where did I put the translation? Here it is. Okay, and look at this. Here it is. Introduction into the investigation of the microscopic structure of vertebrates. It's for beginners. So this is a beginner article on how to dissect animals so that you can put them under the microscope. So it says this, the author says this, unfortunately this topic is not talked about very much for beginners. It gives you a materials list, what you need if you want to cut apart a cat. Really. Stains, even some poisonous stuff, xylene, which we try to avoid. Okay. And yeah, so, and then over here on the next page, it says if you have problems getting all of those substances, then you can contact the publisher. We're going to help you get all of them. So this is like a kind of like a beginner's article on microscopy. And then there's this nice little paragraph here that I just have to share with you, called Killing of Animals. Das Töten der Tiere. It's on page 21 here. And I got this translated for you. Basically, I copy pasted it into ChatGPT and it translated it for me, and I can confirm that the translation is authentic. Okay, so this is how we're going to read this together, just because it's so fun. Fun under quotation marks. To make material procurement as simple as possible, generally only two easily accessible animals, the frog and the young cat, are taken into consideration. Okay, because you want to dissect some animals, let's get some animals that you can easily get. Frog and cat. Killing the animals. That's this one over here. Yeah, down here. Yeah. Frogs are grasped by their hind legs and forcefully struck with their head against the edge of the table. Then the head is cut off with scissors and the spinal cord is destroyed by inserting a darning needle into the spinal canal. I'll tell you a little story of what my biology teacher told me back in the day, because they used to do actually something like this for dissecting. How do you dissect a young cat? Young cats are placed under a glass jar in a cigarette box or another tightly sealed container, and a wad of cotton soaked in chloroform is added. Sulfuric ether is also usable, but cats are more sensitive to chloroform. Once anesthesia has taken effect, the animal is laid on its back on a board. The legs are tied to 4 nails driven in for this purpose, and then the chest skin is cut with a knife, peeled back to the right and left, and the ribs are cut on both sides with the scissors. The cat is still alive. How do you know? Because by removing the middle part, the heart is exposed and the animal is killed by cutting into the heart chambers. A beginner's instructions on microscopy from the year 2021. Okay? We've come a long way. Okay? Right now, luckily, we're having some people are kind of concerned, well, is it ethical to kill microorganisms? And I always say this, well, honestly, I don't have a problem with that because they don't have a nervous system. But honestly, I'm glad that we're thinking in that terms as well, because honestly, this is horrifying. Okay? And I think maybe, why am I telling you this? I'm not only teaching biology in school, but I'm also teaching philosophy. Theory of knowledge, that is, epistemology. And ethical discussions are important. And I think it's also important that we as hobby microscopists know a little bit of aware of where all of our knowledge actually that we have comes from, right? I mean, a lot of animals have been sacrificed in the past. Not only animals, but human experiments and so on. So let's never forget that the knowledge that we have right now, actually, animals had to suffer, still have to suffer for that. I just wanted to share this with you a little bit because I personally found this to be quite, from a historical perspective, a little bit interesting. But I have to tell you, when I read this here, especially with this casual tone, the way that this is written, this is something that shocked me a little bit. And of course gives a little bit a slightly different perspective now when I put some microscope slides of animals or whatever under the microscope, and also Aaron is now mentioning the frog instructions are what I remember doing from my school days. I declined the cat dissection day. I remember being told that the cats were lab cats that were farmed specifically for science. Yes, when I was in school, we were dissecting small fetal pigs. Okay, and my biology teacher told me, this was a long time ago, he said as well, when they were dissecting frogs, what they have done is they basically also did live dissection so that they could actually see the heart beating. Right? So they were actually cutting them open live. So that essentially they could see for educational purposes the organs still working. Nowadays, if you do that, as a teacher, you're in trouble. But this is nothing. How do you say that? When I was at the university, they killed the rats for us, and then 5 minutes later, we were to dissect them. So that's also was a part of also of my education. But for me, I kind of found this text a little bit. See, Aaron also mentioned, yes, our frogs were alive as well, and that was the goal, to see the heart beating. So I'm just saying, times are kind of changing. And I would say, I don't know. It's my personal view. I'm open for debate. I mean, on one hand, I'm glad that we're not doing these things anymore. But on the other hand, I mean, many of us, I myself included, yeah, we have to be careful that we do not become too detached a little bit. I mean, the food that we eat, I mean, how was that food produced? Yeah? So, yeah, very briefly, and now I want to give you a quick look, just because I just want to. These were two sample specimen, and you know what? I don't have bad feelings of actually showing you the contents because all of the contents have been scanned and are available online. Okay, but I consider this to be a very nice and very useful magazine, also for hobby microscopists. Some of the articles are pretty advanced, but if you find an article, and there is an index, also in German, but if you find an article, what I have tried is I simply downloaded the PDF, and if there was something that I was interested in, or that you're interested in, I mean, I understand German, so it's not a problem, but you can actually make a screenshot of this, copy paste the picture into ChatGPT and ask it to translate it. And it will do that. Yeah, taken from a picture. Yeah, so a screenshot. Yeah, you don't even have to copy paste the text. Just take a picture of it, and it works surprisingly well. Yeah, so just wanted to give you a recommendation. The reason is, some of you probably remember that I've been asking the question: how were they actually able to polish down the the spine of a sea urchin? I actually found instructions on that, and it is pretty complicated. Yeah, it's found instructions about this also in one of those magazines. I think from the year 1985. They actually explained how you can actually get a cross section of hard material, and it's pretty complicated. Yeah, you have to carefully grind it down and so on. Yeah. But I just wanted to tell you that the links are in the description below. If you want to have a look, it's a lot of stuff that's available. Yeah, yeah. You just learn German. Yeah, I know German, but it's not an easy language, especially because sometimes the sentence structure is more complex than it should be. Yeah. So next one, let me look at the time a little bit. Yeah, the snake skin. Snakes. Let me quickly have a look. How many people actually voted for the snake skin, and how many for the moose? Let me see. 84%, 38 votes. Okay. Honestly, it's not the most exciting specimen under the microscope, but doesn't matter. This is a snake skin that students have found, actually from two different snakes. Unfortunately, I don't know which ones. The students have found, and they brought it to school. So I'm going to be returning it again, and we actually had I don't know, the whole skin of a snake, huge. And those fragments, I did not rip them off, but actually they just came off and I took them along. And yeah, I thought this is kind of interesting. Let's put it under the microscope. This was kind of the idea. And of course, you know what I'm doing right now. You know what I'm doing right now. Where is this? Here it is. Where is it? Where is it? Where is it? Here we go. Boom. Yeah, so I'm using this USB camera. This is how it looks like. Okay. It doesn't look so much different. Yeah. But the snake skin it's pretty transparent. It's pretty thin. So I don't really, I don't see a reason why we should not be able to make a normal microscope slide out of that. Yeah. So I'm gonna give this I'm gonna give it a simple try. This is actually something that I tried already some time ago. I have to tell you, it is not the most exciting specimen, but it doesn't have to be, right? Because we're exploring and we just want to know how things actually look like, right? So I also tried to do a little bit of staining, but it's keratin, which is essentially protein. And I'm just going to make a regular wet mount. If I can find the tips for my, here they are. I always put the things over here also on the side. Yeah. Luckily, snakes shed their skin frequently. Yep. Oh yeah, here kind of. And I'm just gonna take this thing here, and I wonder if. Yeah, I also recently made a little, how do you say, video about the onion, if you remember the onion skin, and about using not water, but actually cooking oil, not kitchen oil, cooking oil, and for making a mount, because if the specimen is hydrophobic, and maybe this one is as well, then oil would be better, but for our purposes, we're just going to try this here. So we're just going to try to press it as flat as we can. It's going to be folded back a little bit onto itself, but maybe there's a part here that is sufficiently flat. So let me see where the... Yep, here we go. And when you look at this, when I looked at this, it's a little too, but we need to, wrong filter. Here we go. Then we are actually also able to see those textured patterns, and I was kind of wondering, and I don't know, of course, I was not able to see individual cells, but do you see those lines and stripes? And I was kind of wondering whether these kind of are not some kind of protein fibers or not. Yeah. So this is the question that I have a little bit. For me, the structure was rather homogeneous. So, and I did a little research, and it says, it's actually, there are a few cells present as well, but mostly these are, it's keratin, and I don't know whether these are actually, that what you see here, these are actually the individual cells or not, the epithelial cells. It is an animal, obviously, a snake. For this reason, there are no cell walls present, and therefore the individual cells are very difficult to see. Yeah. The cells are so close together, and the cell membranes are so thin. So this is a little bit a general problem with animal tissues, that the individual cells cannot be seen as distinct. And I think that this might have been one of the reasons why it took scientists back in the 19th century so long to actually figure out that also not only plants but also animals are made of individual cells, because you simply cannot see the individual cells because they are not surrounded by a cell wall. Yeah. And so this is a little bit also a something that I've seen over here, where I found this a little bit surprising that it's so homogeneous, so to say, and that there are these fibers that are running throughout. Oxalate crystals. To my knowledge, oxalate crystals are waste. It's waste storage. Yeah. So let's go down a little bit again with the, and let's have a look. You know what? Let's try the phase contrast. If this gives us any additional details, but even in phase contrast, we're only able to see that this snake skin seems to be fairly uniform, and what we have here is these seems to be some kind of bubbles, or how do you say condensed water on the surface. Let's open it again. Those lines are quite quite interesting for me, because this actually means that if you have those structures, those lines that are going through all the way, this means there must be individual cells which have deposited the keratin. How were they able to actually make those lines than if they were individual cells? So that's a couple of things that I'm not quite fully understanding yet. So over here, look, here we are able to see more structures that appears to be like cells, where I'm also not sure whether these are individual cells or just not impressions on the surface. So what I plan to do, and haven't found the time yet, is I do want to experiment around a little bit with different staining techniques as well. Maybe there is a possibility somehow to also increase the contrast of the different structures here of the snake skin. Look here again, we see those round structures which appear to be a little bit, which remind me of cells. Huh? How does a plant get rid of waste? Well, one of the waste gases that plants produce is, well, yes, CO2 is a waste gas as well. Plants also do cell respiration and also oxygen. Yeah. Could be abrasion scratches. That's an interesting point. See, didn't think of that. That actually these lines that you see over here could be scratches in the surface of the skin. Ah, interesting point. Didn't think of that. Did not think of that. For me, I was always kind of wondering how can adjacent cells actually produce a pattern that goes across several cells. This was kind of my how do you say, reasoning behind that. Huh? But the idea could be that these could be maybe some kind of abrasion scratches. Huh? Okay, people. You know what I'm going to do now is I am going to move on. Because it's tradition kind of that at the end I always do a little Q&A session. Yeah, some of the comments I have stretch marks maybe. Yeah. So this is something that I don't know. Yeah. And that's something that one could try to explore. Let's put this carefully back in. Yeah. That maybe some kind of scratches as the snake moves along. One probably would, you know what? That's another thing. Is maybe just to figure out, I think, which one top and bottom part, and then kind of compare those. Yeah, if there are any differences in appearance. Okay. So what I'm going to do now is very quickly go through a couple of your questions or comments. I'm going to go quickly. Okay, so please don't feel bad if I don't. If I don't answer all of those. Could we create a cross-section of a fly or a mosquito? Yeah, but that's elaborate. I have to embed it in paraffin and then have to do a microtoming. Yeah, so this is slightly more complicated. Yeah. Could you glue soft plastic tips to one of your worst pair of tweezers? Ah, you mean that kind of you glue them, those plastic tips maybe that you're able to pick up? Not tried it, yeah, but would be possible. So this is a camera question. I tested 2 cameras, but bad quality on monitor. Any other? Don't get me started because connecting a camera, yeah, connecting a camera to a microscope is a science in itself. Yeah. Honestly, my recommendation is keep on experimenting, and it's not satisfying if I say that. I understand, right? I've had, honestly, camera adapters that worked pretty bad, gave very bad results with one microscope and decent results with another one, which I cannot explain. There seem to be so many different factors contributing that I generally found, and it's not always the quality of the camera, but very often it's the connection of the mount, right? I don't know if it was you, but there was actually one person who once asked, wrote me an email asking, they connected the camera directly, but then you had to completely refocus, yeah, so that the distance between the objective and the specimen were not right again. And this can also deteriorate the quality. So there are so many factors here. What I found out works for me is currently I'm using HDMI cameras for the stereo microscope. I'm using a DSLR camera for my microscope, regular compound microscope, but I'm also putting out an HDMI feed. So you see, I'm mixing all the things together here. Yeah. So this is something where I have to tell you, I don't have an easy answer here. Yeah. Usually you would paraffin embed and microtome the soft tissues. Yes, that's correct. Yeah. I don't know about this. With a large antenna, a male, normally, yeah, because usually the males, they find the females, but yeah, I wouldn't automatically know if this is the case with all mosquitoes. Yeah. These inks are not suspensions; they are solutions, as I mentioned already. I don't think that they're cell organelle specific. Okay, if you want to have highly specific stains, you have to use fluorescent antibodies. Yeah. Do you see fuchsin dye, RT for clothing would stain cells? I mean, I know I've used fuchsin dye. Fuchsin dye is used to stain actually cells. And I've actually bought fuchsin dye, and if it stains clothing, which clothing very often made of cellulose, cotton, which is of course a biological material, then I see no reason why it should not. Of course, but fuchsin actually is a documented stain that is used for staining cells. And I once did some research. How did the early microscopists actually figure out the whole thing with the stains? And indeed, what they have done is it was actually the other way around. They actually talked to people who were staining cloth, and they experimented with those stains. So they actually used stains that were used in the clothing industry. Yeah. I wonder if mixing the ink with alcohol made it better on the cells. Yeah, next experiment. Okay? Could be. Yeah. Das große Kosmosbuch der Mikroskopie, I've got that. A black suspension can be used to make the mucous surroundings of algae or bacteria visible. That is interesting. The mucous surroundings. The mucus, I can imagine because maybe the mucus will catch the black particles. Maybe the whatever black particles are in there, maybe will stick to the mucus, and therefore it will make it. Yeah, I've got that book, but did not read that chapter yet. You never label anything. Who are you, and where is the real Oliver? Story. You have to. Let's put it this way. When I do demonstrations here in the live stream, I try to keep it a little bit time efficient. And I want to demo so that, and in many cases, or in some cases, I actually throw out the things that I'm not going to be using anymore. So, but actually, what you have to do is you have to label everything if you want to keep it. I even bought myself a little notebook here. Yeah, to keep notes, but you got to be consistent. I admit it. Yeah, that's the thing. Yeah, that's what I also suppose. The suspended particle stick. Yeah, the chloroplast might have a protection layer, not letting suspended particles in. Yes, the particles are large, and if they're not able to penetrate the cell membrane, then yeah, they're going to stay out there. Yeah, most soluble dyes have a negative charge. Okay. Might be worthwhile to measure the pH. I don't know how to do that. I need to have a very fine. Well, the point is with litmus paper, it's not going to work because it's going to stain the paper. Yeah. Yes, I've done that. You remove the stain. Okay. Yeah, the frame rate. The second look at the turquoise specimen. I think I already put it away. Sorry. Yeah, it's already stuck together with another slide. Yeah. Okay. Oh, that was it already. Good. Let me quickly have a look at some of your comments here. Page 269. Tintenausstrich. Okay, I will have a look. Thank you very much. I will give it a look here. I'm still looking at some of the comments here. Do you have experiments with entelan? No, I don't. I don't have experiments with that, and I also do not do fluorescent imaging. I have to admit, so because you need fluorescent antibodies, and that's a different. How do you say? It's a different price category. Yeah. Keeping a recycling container for temporary slides, if I remember correctly, a recycling container. You mean a container where I drop in my used slides? I show you something. Look what I got here. Yeah, this is the place, and that's the one, the stains that I've just done. See, it sticks together. So what I usually do here is nothing dangerous in here, right? So nothing infectious or so. So I pour hot water over it, allow it to dissolve, and then I clean each slide separately. Yeah, so that's, yeah. And then I check a little bit against the light whether they're reasonably clean. Okay. Can you make an introduction video about using cameras with the microscopes, how to customize it a bit more, maybe some terminology as well? That is a yes, but that is a large topic. I will tell you, this is connecting a camera to a microscope can be a frustrating thing. Doesn't have to be, because sometimes there is no solution that's 100%. I mean, there are some solutions that work really well, like the one that I have on my microscope over here. But that is basically a complete solution out of the box, and it's a cost issue, right? So that is, and there are those how do you say, standard adapters, DSLR adapters that you can get. Some of them work better, some of them don't work better. So there is some microscope allow you to connect C-mounted cameras, others do not. It's a bit frustrating. So what many people or some people do is they try to find their own solutions by actually making their own adapter. Yeah. So this is resolution of even the best objectives are much less than the camera. Problem is not the cameras. Yes, I totally agree. I get a lot of comments, emails, what is the best camera to use? And I say, it's not the camera. The cameras are way better than what the microscope is able to deliver. Yeah. So that's the thing. And with a DSLR, like the 380T, you'd likely want intermediate optics. Yes. Yeah. But those intermediate optics can also degrade image quality. So you need to, yeah. So you see, it's all a little bit of a trial and error. Yeah. So I'm quickly going to go through. I also have to be a little bit time considerate. Do you have any videos where you explain how to do DIC on a microscope? No, I don't have a video on that. And the simple reason is because DIC, differential interference contrast, there is no homemade solution for that. You need, and you cannot simply upgrade a microscope to DIC unless the microscope is designed to do that. And if you want to have DIC, the company Euromax offers DIC, and also the other big four companies like Olympus, Leica, Zeiss, yeah, and and Nikon, they also offer DIC, but there is no possibility to do a simple upgrade. And I probably would say that I mean, I like to use DIC, of course, because I like to share the videos and they're nice and colorful. But in the area of hobby microscopy, there are so many things that you can do without that. Yeah. Let me quickly go through it. Otherwise, there are no. What is the purpose of those oxide crystals in the onion skin? What I read somewhere is that they can be used for storage, but also as waste. So very often it's also a way of storing waste, so that they do not interfere with the metabolism of the cell. Yeah? The most important thing that I would point out with such slides is that they're important for medical research. Without them, we wouldn't be able to treat the animals as well. Yes. Yeah, and it doesn't always mean that those animals, like the monkey's tail, that they have been using animal cruelty, right? So one doesn't automatically have to assume that. But I have to admit that this article that I just showed you before was a little bit surprising to me as well. Yeah, yeah, and then the question about Reinberg filters, which I basically already covered. Yeah, so I do hope that I think enough for today. What I'm going to do now is the following. In my other channel, I'm going to now make available this newest explainer video. So in this Microbehunter channel, not this one, but the other channel. Have a look at it. I'd like to hear your feedback. It's a little more on the I wouldn't say academic, but more on the less casual. Right now we're having more or less a casual conversation. The other video is a little bit more scripted. I'd like to hear your opinion about that. It's again for a slightly different target group maybe, but like to simply also hear your opinion. So I'll put it online right after the live stream here. So what I'm going to do, however, for today is I'm just going to call it quits for today. I want to thank you that you joined me again in this in this live stream. I hope to be able to see you again next week. And I think that's pretty much all that I have. If you have a couple of nice pictures that you would like to share with the community, please send them to me per email, and I will also make them show them in the live stream. Okay. Do you clean the slides using your bare hands? Yes, because I don't have anything dangerous on the slides. Yeah, and I pour hot water over it to kill off any microorganisms. Yeah, so that's the thing. Yeah, so enough for today. I wish you all the best. Happy microbe hunting as always. Thank you that you joined me this week again, and hope to see you again. Bye bye.